Thanks again from the team of Open Block Science. Thanks, Jennie and Jerry. So today, this is a great opportunity to present my work to the all scientific community that we have recently published in a Science Translational Medicine. But it's always nice to talk more in person about your work than just through the papers. So in this work, basically, we demonstrated that the thalamic reticular nucleus restoration of activity improves sleep architecture and reduces A-beta plaque deposition in Alzheimer's disease mice. If you all know that Alzheimer's disease is associated with the cognitive impairments. So from the start of the diagnosis of Alzheimer's disease to the learning and memory deficit, there are several events happen during the disease, but still need to be elucidated. And we don't know how the path from the Alzheimer's disease to those symptoms like learning and memory deficit happens. And one of the candidates so mainly it's because of, so we know from the various familiar forms of Alzheimer's disease that we could basically understand the role of amyloid beta. And there are these familiar forms of AD like presenting mutation, APP mutation, APP duplication that helped us to design various transgenic models based on that and basically study how the A-beta plays role in the development of disease and leading into the learning and memory deficit. So throughout about 20-30 years of work we have done in Alzheimer's disease, and most of the work has been hippocampus-centric. But the diversity of this disease and complexity cannot be explained just by the hippocampus-centric view. So we started looking into another major network system that's called corticothalamic network that basically shows the communication, reciprocal communication between cortex and thalamic relay. That basically processes all the afferent information coming to the brain. And in between that, there is one nucleus called thalamic reticular nucleus, which is basically regulatory nucleus, controls this communication between cortex and thalamus. We know in Alzheimer's that many of the symptoms like memory disorders, mood disorders, and occurrence of some non-convulsive and convulsive seizures that's been now known in Alzheimer's are regulated by hippocampus. However, there are some other symptoms that's commonly exhibited and subclinically reported as well, are sleep maintenance and decrease in slow wave sleep, as well as attention deficit and occurrence of some non-convulsive seizures. So these symptoms are basically regulated by this thalamocortical network. Basically, that's what I've shown here. And this TRN, which regulates this communication between cortex and thalamus, we came up with the question whether this TRN is a master regulator of cognitive and behavioral impairments in AD. So we started, not that we started, but my PI who used to work for Dr. Len Muke, who designed this J20 mouse line, and we've been using this mouse line for several years, maybe a decade. This mouse line shows a faster progression of disease due to the double mutation that includes Swedish and Indiana mutation carrying the human mutant APP. And this mouse line exhibit A-beta production at the start of the birth, which is at measurable level from the age of 1 month. And you start seeing the plaque deposition at the age of 6 months. In between this time, there is a synaptic loss at the age of 2 to 3 months and cognitive deficit by the age of 3 to 4 months. And So we are really interested in what happens before the plaque deposition, like what predisposes this. And we are really interested in this age from 1 month to 4 months. That's when particularly we see the occurrence of epileptiform activity, seizures, slip fragmentation, and decrease in slow sleep. So it all started back in 2016 when our group first published the data where they demonstrated that this normal TRN, normal corticothalamic circuitry, which is basically exhibited dysfunction at the level of TRN activity, which cause, so decrease in TRN activity resulted into disinhibition of thalamic relay, so increase in thalamocortical response. So we have to know that the TRN is completely GABAergic population, and it basically provides the feedback inhibition to thalamic relay. And if the disinhibition of thalamic relay could lead into hypersynchronous activity and could be possible effect onto the occurrence of non-convulsive seizures usually observed in absence epilepsy, as well as lack of sleep maintenance by increasing sensory afferent information to the cortex. So during this study, our group basically tested TRN in APP and NTG. NTGs are the control non-transgenic mice. And what they found in TRN, that the delta phosphate, that's the activity-dependent marker, exhibited dramatic reduction in activity. So basically, delta phosphate is one of the forms of phosphate, which is the activity-dependent marker and shows the accumulation upon stimulation, and which stays in the system for several days. So this basically, I can simply say that the delta phosphate exhibit the chronic activity of neurons, and that can be tested by immunohistochemistry. So they found this strong reduction in delta phosphate immunoreactive cells in TRN in APP mice, and that's at the age of 2 months. And they were wondering whether is it because of neuronal loss, but there is no neuronal loss apparently. So the neurons are still there, they're just not active. And then obviously the next task was to look into how the decrease in TRN activity is affecting sleep in this mice. So they tested sleep in this J20 mice at the age of 2 months using the home cage scan technique, which actually they allowed them to test whether mouse is sleeping or awake. And what they found is the mouse spent significantly higher number of times in awakening and less time in sleep in comparison to their non-transgenic control. When I came, I was first worried whether this is just the mouse line issue, whether it's only in a J20 mice. So we tested other mouse line as well, and we tested TG2576 with equivalent age, and also in APP-PS1 mice, and we found both the mouse line demonstrate the same reduction in activity of thalamic reticular nucleus. And when we tested in I5 mice, which basically I5 mice show overexpression of APP, but here that's the healthy, when I say wild type human, means the healthy human APP expression, which does not lead to A beta production, which is contrary to J20 mice, which express the mutant APP. So we found that the accumulation of A beta, overproduction of A beta, does not relate to the delta reduction in activity of TRN, and been shown with the I5 mice. So with all that, we came to binding these three elements. That's one, the thalamic reticular nucleus, sleep and Alzheimer's disease. Thalamic reticular nucleus is important for sleep maintenance and for slow wave activity and induction of sleep spindles. It's been known in relevance to the Alzheimer's and dementias that the slow wave sleep is important for memory consolidation, clearance of, as well as clearance of waste material from the brain. It has been studied in Alzheimer patients that says that this decrease in the patients, as well as if you disrupt the slow wave activity in a healthy subjects, they found increase in A beta production in CSF. So it's also been known that the sleep fragmentation or the poor sleep predicts risk 8 to 10 years prior to the onset of dementia symptoms. So it is the great point where we can start focusing that the sleep might be majorly responsible for this culmination or the worsening of the disease, and that can be targeted several years before the diagnosis of Alzheimer's. So I just would like to show a small clip from Dr. Jeff Phillips' TED Talk. This where they first time exhibited that the glymphatic system helps in clearance of metabolites from the brain during sleep. So what they did with the multiproton imaging, that they recorded in vivo activity in the brain while injecting green tracers when mouse is awake, and then the red tracer when mouse goes to sleep. Let me see if it works. Last time it didn't work somehow. Oh, my apology. But basically, I can explain this part. Is when mouse goes to sleep, they saw flush of cerebrospinal fluid through the brain, and basically they recorded with the ray tracer. That basically says that during deep sleep, our brain starts cleaning, and that's with the help of the glymphatic system. And that could be crucial in perspective of various neurodegenerative diseases, including Alzheimer's, where we have accumulation of A beta and tau, and that leads to various pathophysiological changes in the brain. And sleep could help, deep sleep could help in clearance of those metabolites as well as the toxic material from the brain. So initially, looking at this point, we were interested in looking at whether this sleep fragmentation, whether it's associated with plaque deposition in FPP mice. So I used the same data that we collected with home kit scan prior to starting with the EEG analysis. So I was just interested in looking at the A beta plaque and awakening correlation, and this is in a 6-month-old mice. And I found that in cortex and hippocampus, there is a positive correlation between more the number of awakenings, there is a more A beta plaque deposition in our mice. So it brings all this together, and then we were actually interested in doing a more physiological way of recording sleep in our mice. So for that purpose, we use this the system that's been created by Data Science International. It allows us to record EEG and EMG activity in mice. Basically, it's a wireless system that helps helps mouse to move freely in its home cage. So the first part was to assess the sleep using a telemetry system in our J20 mice. So I tested that first in our 2-month-old mice, and this is the sleep in non-transiting mouse control. What we see here is the sleep hypnogram. That's active wake, the green, quiet wake, REM sleep, and the slow wave sleep. Basically, the red line indicates the slow wave sleep. We could categorize different stages of sleep using EMG, that's recorded from the neck muscle, and EEG from the frontal lobe. With the help of EEG, I could calculate the delta ratio and theta ratio. Which basically delta ratio is the mark. Delta ratio one can use to classify the slow wave sleep, and theta ratio for the REM sleep. So slow wave sleep is a part of a non-REM sleep. And the one thing is need to be noted here that the mice are not like us. They are nocturnal animals. So they sleep during the day when the lights are on, and these are active when the lights are off. So this thing was done from the side giver 0, that's the 7 a.m. to 7 p.m. in the morning. Now, if you look at the representative APP mouse, what we see here that the APP mouse woke up more often in comparison to the NTG mouse. That simply says that here that the APP mouse here is exhibiting the sleep fragmentation. And we quantified that data in our 2-month-old mice first. What we found that the APP mice awake twice the number of times during the day cycle, during their sleep period, in comparison to a non-transgenic control, and they spend more time being awake. If we look at their slow wave sleep, we found that there is a decrease in the amount of time they spent in a slow wave sleep, that is part of non-REM sleep. However, we did not see any changes in REM sleep, but that's also because we saw so much variability among our APP mice. So, and overall, there is a decrease in total sleep that APP mice had during the day cycle. We were interested whether this starts even before 2 months. So we tested in 1-month-old mice, and we saw that it was evident that they had sleep fragmentation starting from a 1-month-old. So we came up to the question, the TRN dysfunction, how we can solve that in Alzheimer's disease. So TRN exhibited a decrease in activity. So the one way we could restore the activity in TRN was using the chemogenetic method. So if you know that the DREADs are basically designer receptors exclusively activated by designer drugs. So obviously, can we just restore the activity of TRN? So, and if we can, we will be able to basically solve all of this problem or some of this problem, including sleep fragmentation, reduction in slow wave sleep. Basically, that leads to decrease in A beta clearance and ultimately responsible for memory deficit. And also some forms of epileptiform activity. We haven't yet digged into that part, but TRN known for TRN dysfunction is known for an induction of spike wave discharges in Upson's epilepsy. So that would be interesting paradigm that we are interested in looking at too. So first we started with targeting and activation of TRN using DREADs. So as I had mentioned, that the TRN is made up of whole GABAergic population. So we crossed our NTG APP mice, that means J20 mouse line, with the GAT2 high-risk mice, so that we can target our CRE-dependent, not our CRE-dependent HM32 virus directly into TRN. So that's what we did. So this virus we basically injected bilaterally in TRN and obtained this expression that was beautifully localized in a thalamic reticular nucleus that you can see with the stained with the M cherry. That's basically the reporter gene. And the next part, we wanted to check whether the activation of TRN. So if you know that the HM32 DREADs works you can express those viruses. Basically, the HM30Q is GQ coupled receptor, and you can activate that only by inert compound that's been debated for a while. That's basically clozapine oxide. So what we did to basically test if we are able to activate TRN, we tested a group of mice that had been injected with saline and a group of mice injected with CNO. And for in vivo validation, we obtained their brains and stained with the delta phosphate again. And where you can see here, the mouse brain that's the mouse that been treated with saline shows expression of phosphate delta phosphate with overlapping expression of HM30Q. And in CNO, we can see there is increase in phosphate delta phosphate expressing cells. Those are the green cells. So it basically says that we could activate TRN, and that's first we tested in NTG mice. On the next part, we wanted to see if we can see the same effect in a slice physiology. So we performed slice physiology with our collaborators from across the street from the UT Health. And where Fred, he recorded from NTG mice that have been injected with HM30Q M-cherry virus in TRN, as well as in thalamic relay. In TRN, after application of CNO, he could see action potentials within a few minutes in slices. And when he recorded in thalamic relay, after bad application of CNO, he could see the inhibitory responses. When he checked the same in NTG M-cherry mice, those are basically the negative control, they don't express HM30Q. He did not see any action potential. So it kind of, we validated that the CNO does not elicit any action potentials or inhibitory responses non-specifically. Then the next part, we wanted to obviously go on and check if we can use this method, use this system to restore activity, restore sleep in our APP mice by restoring activity of TRN. So we tested first in 2-month-old mice. The same method, we injected this HM30K M-cherry virus bilaterally in TRN of APP GAT2 mice and also NTG GAT2 mice. And we received the expression after 3 weeks of virus expression, virus infusion. So for the treatment protocol, basically we started with saline injection at a start of day cycle. So this saline injection would give us the baseline of like every mouse sleeps differently, just like us. So we needed to get its own baseline. So all the mice received saline injection at the start of a day cycle on day 1. And on a day 3, a group of mice received saline injection and a group of mice received CNO injection. Throughout this time period, we performed telemetry recording like I shown in the last figures. What we found after that acute activation of TRN using single dose of CNO, that with the CNO treatment, we saw increase in slow wave sleep in our APP mice and decrease in number of awakenings. So if you see this is basically first 6 hours after treatment. So the treatment started at the site gave 0 at the 7 a.m. in the morning. And this is on a day 1 as its own baseline, and we saw increase in number of awakenings as usual we saw in APP mouse. And on a day 3, we saw a strong reduction in number of awakenings. And you can see here how the slow wave sleep is so smooth and less interrupted in comparison to the day 1. When we quantified this data and compared with other groups, we saw that NTG HM30Q, that mice that have been treated with saline, show significant difference in comparison to APP mice, HM32 that have been also treated with saline. So basically the control groups. And if the HM32, APP HM32 mice are treated with CNO, they exhibited normalization of number of awakenings that was similar to the NTG group. And we saw this effect during the next 6 hours of the daylight cycle as well. When we looked into the slow wave sleep, we saw the slow wave sleep significantly increase after CNO application in an APP HM32 mice during first 6 hours. So the next part we were interested in looking at was how this improvement in sleep will help clearance of A beta plaques in APP mice. So like we have seen, the glymphatic system works during the deep stage of sleep. And to effectively help in clearance of A beta, we need to activate TRN in a way that the sleep will be restored throughout a time period, because the acute treatment perhaps will not have that strong effect on the plaque deposition. So for that, we wanted to test a chronic activation of TRN, where there would be a long-term treatment that could help TRN to stay active during certain time, and that could help also improve restoration of sleep in this APP mice. So we tested the same way by injecting HM32 virus bilaterally in TRN. But this time in a 6-month-old mice, that just the time before they start showing A beta plaque deposition. Similarly, we allowed the virus to express for 3 weeks, and we obtained the baseline by injecting all mice with the saline at the start of a day cycle. And then for the next 30 days, a group of mice received saline injection and a group of mice received CNO injection at the start of a day cycle every day. And every 5th day of the treatment, we obtain EEG and EMG recording to test whether it is improving sleep in mice. So let's look at the sleep, how it has an effect after chronic TRN activation. So what we see here is this the baseline sleep after saline treatment in APP mouse. Like we have seen with the acute treatment, even in 2-month-old, even in a 6-month-old, we saw this strong reduction in number of awakenings and increase in slow wave sleep during first 6 hours. Here I am showing only first 3 hours. And now if we look at the day 10, 20, and 30, we see that it consistently improved sleep in our APP mice throughout the treatment. And when we quantified this data, which shows the first 6 hours of analysis, so the red dots are basically APP mice who are treated with CNO. And if you compare that with the pink ones, those are the control APP mice. We see that consistent robust reduction in number of awakenings in the CNO treated mice. As well as we see that increase in slow wave sleep in the after CNO treatment, after chronic activation of TRN. We did not see any changes in REM sleep and also in total sleep overall. After that, we received brains from these mice, and we wanted to test how the plaque deposition looks like after CNO activation of TRN in CNO treated mice. What we see here is that's really early stage of plaque deposition. These are about 7-month-old mice. And what we see, the A beta plaque deposition in a hippocampus as well as in cortex, and this is the saline mouse. And if we compare that with CNO, we see a strong reduction in plaques, number of plaques in CNS-treated mice in cortex as well as in hippocampus. And with the after quantification, we see that after 30 days of TRN activation, we saw about 30% decrease in area covered by A-beta plaques in these 6 months, 7 months old mice. That's sure and fine. So what's the situation in the patients? Obviously, we want to see how it would be effective in the patient. So we wanted to start with checking, are the patients also, 80 patients also showing this TRN dysfunction. So we collected brains from NIH Neurobiobank. We collected brains from the 80 patients as well as age-matched control and few mild cognitive impairment patients as well. And what you see here is the control, age-matched control patient. And this is the TRN showing the delta phosphate expression. And this is the ventrolateral nucleus of thalamus, basically that receives the projections from the TRN and communicates reciprocally with it. And if you look at the MCI patient, and the AD patient, we see in the AD patient, there is a strong reduction in delta phosphate expressing cells. We can see that in higher magnification and insight that the AD patient had fewer delta phosphate expressing cells. Perhaps that suggests that there is TRN dysfunction also present in AD patients as well. And what we see here in the parentheses is the Braque stage, which demonstrate the severity of tauopathy. 0 is none, to the 6 is basically the severe stage. And if we do the correlation analysis with the delta phosphate TRN cells and the Braque stage, we saw if there is a fewer delta phosphate expressing cells in TRN, they have higher Braque stage. That could indicate basically that the decrease in TRN activity has indirect effect in tauopathy and could be related with a lack of sleep maintenance and any kind of sleep fragmentation that could be associated with clearance of tau protein. Also, the same way we wanted to test, like we tested in our mice, if there is a neuronal loss in AD patients. Apparently, there was no neuronal loss in TRN when we checked into their in 80 patient samples, and we did not see any correlation with the neuron positive TRN cells and delta phosphate TRN cells. So it does indicate basically that the cells are there, the TRN cells are there, they are not dead, but they exhibit decrease in activity even in the patients. So with that,
hi Rohan, thanks for the presentation. Due to time constraint, could you maybe give us a wrap up for about 1 minute for your project? Thanks.
Yeah, sorry about that. So basically what we see here is that the TRN hypofunction could lead into sleep fragmentation and decrease in slow-wave sleep and A-beta accumulation. If we restore the activity of TRN, we could able to normalize sleep in APP mice, and perhaps we'll be able to improve symptoms and modify disease progression. That's it. Thank you. Sorry, I think I ran over the time. Thank you, and I'm ready for the questions.